Hi, my name is Eric Armstrong. I'm a PhD candidate at the University of Toronto. I'd like to first thank the organizers for inviting me here to speak today. And the title of my talk is The Impact of Lactin-V on Genital Immunology Following Standard Bacterial Vaginosis Treatment, Results from a Randomized Placebo-Controlled Trial. The vaginal microbiome is an important determinant of HIV risk among women and refers to the endogenous microbes of the female genital tract. It's typically composed of either one predominant species of lactobacillus, like Lactobacillus crispatus, or a diverse population of anaerobic bacteria, also known as bacterial vaginosis, or BV. Now, BV has been consistently linked to elevated HIV risk among women, which we believe is due in part to the induction of genital mucosal inflammation. And genital inflammation increases HIV risk through two main mechanisms. The first is through the disruption of the epithelial cell barrier, which can be done directly by pro-inflammatory cytokines like IL-1 alpha. And what this does is essentially makes it easier for HIV to cross the epithelial cell barrier and reach target cells like CD4 T cells on the other side. Second, genital inflammation induces the recruitment of HIV target cells to the mucosa, thus increasing more opportunities for an infection to occur. Now, it's typically difficult to directly evaluate epithelial barrier integrity in vivo in the human female genital tract, but there are some more indirect ways of doing so, like measuring the soluble form of membrane proteins in secretions. One such membrane protein is E-cadherin, which is a component of the adherens junction, which essentially just acts to hold together epithelial cells. But when the epithelial barrier integrity is compromised and E-cadherin is cleaved, we can measure the resulting soluble fragment of E-cadherin, also known as soluble E-cadherin, in secretions. So in other words, when there's high soluble E-cadherin, there is a high epithelial barrier damage. Now, BV is typically treated with oral or topical antibiotics like metronidazole, but recurrence rates are unacceptably high, reported at about 40% at 3 months post-treatment, indicating the need for better treatment options. But fortunately, alternative treatment strategies are being actively pursued. Our collaborators, Drs. Craig Cohen and Anke Hemmerling, among others, recently published findings from a phase 2b randomized placebo-controlled clinical trial looking at the impact of a novel L. crispatus-based live biotherapeutic, also known as Lactin-V, to prevent BV recurrence following standard antibiotic treatment. The primary finding from this study was that at week 12, or about 1 week after the last dose of Lactin-V or placebo, there was significantly lower BV recurrence in the Lactin-V arm compared to placebo. But interestingly, at week 24, or about 3 months after the last dose, there was still significantly lower BV recurrence in the Lactin-V arm compared to placebo, suggesting that there may be sustained benefits to Lactin-V. So our objective in this study was to investigate the sustained impact of Lactin-V on genital immunology in the vaginal microbiome. So the larger clinical trial enrolled 228 women with BV, provided them with topical metronidazole at baseline, and then at 1 week immediately following the end of antibiotic treatment, randomized them 2-to-1 to receive either Lactin-V or placebo, which was self-applied for 11 weeks, ending 1 week prior to the visit at week 12. I've also highlighted our primary analyses here at 24 weeks. Vaginal swabs were also obtained at each of those time points, which we analyzed for soluble immune factors and the absolute abundance of some key bacteria species, including the 4 most common vaginal species of Lactobacillus and 4 common BV-associated bacteria. Metagenomic analyses were also performed at the Chan Zuckerberg Biohub, which won't be shown today. We performed these assays on samples from 48 randomly selected, highly compliant users who used at least 75% of their doses, and these participants remained in the 2-to-1 randomization. In addition, all immune assays were performed by blinded lab personnel. First, we compared L. crispatus absolute abundance between the Lactin-V and placebo arm at week 12 and week 24, and we see that there's significantly higher L. crispatus in the Lactin-V arm at week 12, which was sustained to week 24. The y-axis on this figure shows the absolute abundance of L. crispatus measured in cycle threshold, or CT, and I've reversed that y-axis because a low CT reflects high absolute abundance. Next, we wondered what the immune implications of this increase in Lactobacillus crispatus would be. So we looked at the correlation between the change in L. crispatus absolute abundance from baseline to 24 weeks, shown on the x-axis of both these figures, and the change in IL-1 alpha and soluble E-cadherin from baseline to 24 weeks, remembering that high soluble E-cadherin reflects high epithelial barrier damage or loss of epithelial barrier integrity. And we see in both figures that there's a significant negative correlation between the two, suggesting that the increase in L. crispatus is conferring lower levels of inflammation and and improved epithelial barrier integrity. However, we already know that BV-associated bacteria like Prevotella are strongly correlated with inflammation in the female genital tract, suggesting that they could confound this relationship between crispatus and immune factors. So we then looked at the relationship between the change in Prevotella from baseline to 24 weeks and the change in IL-1 alpha and soluble E-cadherin, and we see that in both cases, Prevotella is a much stronger predictor of both of these and is very strongly positively correlated with both IL-1 alpha and the change in soluble E-cadherin from baseline to 24 weeks. Next, we wondered what the possible mechanisms for which L. crispatus was conferring lower levels of IL-1 alpha and soluble E-cadherin could be. So we looked at the correlation between the change in L. crispatus absolute abundance from baseline to 24 weeks and the change in Prevotella absolute abundance from baseline to 24 weeks. And we see a negative correlation between the two, suggesting that these immune effects associated with crispatus may be due to the exclusion of BV-associated bacteria like Prevotella. To summarize, Lactin-V induced a sustained increase in vaginal crispatus for at least 3 months after BV treatment, and this increased absolute abundance of L. crispatus was associated with improved epithelial barrier integrity and reduced inflammation. In addition, we believe this immune impact may be due to direct effects and/or the exclusion of BV-associated bacteria. In addition, current studies are actively investigating the impact of Lactin-V on HIV risk among young South African women, which is very exciting. I'd first like to thank my supervisor, Dr. Rupert Call, and the rest of the Call Lab. I'd also like to thank our clinical collaborators at the University of California, San Francisco, Drs. Craig Cohen and Anke Hemmerling, and I'd like to thank our funders at the Canadian Institutes of Health Research and the National Institute of Allergy and Infectious Disease at the NIH. Thanks very much.